The human brain contains approximately 100 trillion synaptic connections. Mind uploading requires mapping this entire connectome, the complete wiring diagram of neural pathways. This map must capture the precise location and strength of every synapse, defining the physical substrate of memory and consciousness. The challenge is fundamentally one of resolution and scale. We need nanometer precision across a volume of over 1,000 cubic centimeters. Current technology struggles to map even a cubic millimeter of mammalian cortex completely. Achieving whole brain emulation depends entirely on overcoming this immense data acquisition hurdle. Structural connectomics relies primarily on Serial Section Electron Microscopy, or SSEM. This technique involves slicing fixed brain tissue into ultra-thin sections, typically 30 nanometers thick. Each slice is imaged individually using an electron beam to reveal cellular membranes and synaptic clefts. A major technical hurdle is maintaining tissue integrity and preventing distortion during the slicing process. Furthermore, aligning tens of thousands of two-dimensional images back into a coherent three-dimensional volume introduces significant computational error and requires sophisticated registration algorithms. Manual tracing of neuronal processes is prohibitively slow. A single human researcher takes years to map a small fraction of a mouse brain. Automated reconstruction is mandatory, utilizing machine learning algorithms to segment neurons and identify synaptic junctions. These algorithms analyze image contrast to distinguish axons, dendrites, and glial cells. However, machine learning output requires extensive human proofreading, often called neurosurgery by committee, to correct errors in tracing and misidentified connections, especially in densely packed neuropil. The scale disparity illustrates the difficulty. The nematode worm Caenorhabditis elegans, the only organism with a fully mapped connectome, possesses exactly 302 neurons and 7,000 synapses. The human brain contains 86 billion neurons. If mapping the worm took over a decade, scaling that effort up to the human level, which involves 100 trillion synapses, demands a technological leap equivalent to moving from the abacus to quantum computing. Current projects focus on small mammalian volumes, such as the mouse visual cortex, as intermediate milestones. A structural map alone is insufficient for functional replication. Synaptic strength is dynamic, modulated by recent activity and biochemical state. The connectome map captures the physical architecture, but not the functional weights. Neuromodulators like dopamine and serotonin alter neuronal communication globally without changing the physical wiring. A complete upload requires capturing the functional state, including the concentration gradients of thousands of proteins and messenger ribonucleic acid molecules at the moment of scanning. Capturing the dynamic state necessitates freezing the brain instantly to halt all biochemical processes, a process known as vitrification. This must occur without introducing structural artifacts that distort the tissue geometry required for SSEM. The challenge is achieving uniform freezing across the entire human brain volume, preventing ice crystal formation which destroys cellular structures. Current vitrification methods are effective only for small tissue samples, not for large, dense organs. Achieving artifact-free whole brain vitrification remains a major physical engineering barrier. The data storage requirement for a full human connectome is staggering. Estimates suggest that capturing the necessary nanometer resolution data, including both structural and functional markers, would generate between 1 and 10 zettabytes of raw image data. 1 zettabyte equals 1 billion terabytes. Processing and storing this volume requires infrastructure far exceeding current global data centers. Furthermore, developing standardized formats for connectomic data sharing remains an unresolved issue, hindering collaborative research efforts and data validation. If successful, mind uploading raises profound questions regarding personal identity, a core concern in secular humanism. The process involves creating a digital copy based on the physical substrate. If the original brain is destroyed during the scanning process, does the digital instantiation retain the original consciousness, or is it merely a perfect replica? This philosophical problem, known as the teleportation paradox, challenges the continuity of self and the definition of life beyond biological constraints, forcing a re-evaluation of what constitutes personhood. Current progress focuses on partial mapping. The Drosophila fruit fly brain, containing approximately 100,000 neurons, is nearing completion. Researchers have successfully mapped the mouse retina and small cortical columns. These efforts validate the SSEM pipeline and the automated reconstruction tools. However, scaling these methods linearly to the primate or human brain is infeasible due to the exponential increase in complexity and the required imaging time. New high-throughput imaging modalities are required to accelerate data acquisition by several orders of magnitude. Whole brain emulation, or WBE, remains the ultimate goal, but alternative strategies are emerging. Partial connectome mapping, focusing only on critical memory centers like the hippocampus, might offer limited functional restoration. Furthermore, synthetic biology approaches aim to build biological neural networks from scratch, bypassing the need for scanning entirely. The pursuit of WBE drives fundamental advances in neuroscience, data science, and high-resolution imaging, pushing the boundaries of post-biological existence and redefining human potential.